I am Diego Rubiales. I am professor at CSIC and the Institute for Sustainable Agriculture at Córdoba. And I have been working in legume breeding mainly for disease resistance in the past 25 years. Before, I was working in cereals. And in this particular project, I am co-leading the work package 3, that is the development of adjustment of new phenotyping tools, and also contributing significantly in other work packages, mainly on genetic analysis, phenotyping, and use of mapping on different crops and genomic selection. In particular, I am also crop leader of lentils, that is this poster, and also on veg. But we participate, in my institute, we participate also strongly in fava bean and in pea activities. In this particular poster, I was responsible to develop as I am crop leader. So we are listing here the activities performed in the first year and what is planned for the coming 3 years. And lentils is particularly interesting crop. Spain is the largest producer in Europe. We are the largest consumers, but there are not particular breeding programs. So then we realized in the project there is an increasing interest in almost all European countries in developing lentil research, but surprisingly there were no capabilities, little expertise, little resources, or little activities go on. So mainly the activities are restricted to us, CSIC, and ICARDA. Also in French, in RAD, we will do a little bit on genotyping. And what we are doing is we are sharing and testing in the field a panel of about 300 lentil accessions already genotyped by ICARDA, and we are phenotyping under field conditions for yield, a number of agronomic traits, and a number of diseases. We are also phenotyping under control conditions for a number of diseases, and then with this we will develop GWAS analysis to identify the markers associated with the traits and use a genomic selection model. I have been involved in quite many projects on legumes, both national and international, but in this particular one, I am happy because we are just not intending to do very deep research by itself, but to translate the results to the sector, to the plant breeding companies that are here. So we are not trying to answer deep complicated scientific questions, but just to resolve the problem that they told us. So they told us, we have this problem with this crop, we don't know how to screen, we don't have resistance to this, and then we are trying to address exactly those concerns that they told us that they are important for them. Well, particularly I am in the end of my career, so that is what I want to do. I already published quite substantially in the past, and I will continue publish, but I want to see the outcomes of this translated to the plant breeders, to the farmers, and this project I think is quite well focused on that.